Hey, Mr. P here. In this video we're going to talk about meiosis and genetic diversity, so specifically objectives D219, D2110, and D2111. In our past videos we've talked all about cell division, we've talked through mitosis, we've talked through DNA packaging, we've talked through a lot of things that have led up to meiosis and how meiosis results in genetic diversity. And so if we look in more depth at the objectives, we're going to notice that D219 specifically calls out meiosis. Students should understand the terms diploid and haploid and how the two divisions of meiosis produce 4 haploid nuclei from 1 diploid nucleus. They should also understand the need for meiosis in a sexual life cycle. Students should be able to outline the 2 rounds of segregation in meiosis. Again, meiotic division requires 2 rounds of reduction division. We're going to talk about that in more detail as the video progresses. D2110 talks about Down syndrome and non-disjunction. Now, if there is an error in meiotic division, it will result in a syndrome known as Down syndrome, which happens as a result of a non-disjunction event. Again, we'll talk more about that. And D2111 talks about meiosis as the source of variation in sexually reproducing organisms. So, students should understand how meiosis generates genetic diversity by random orientation of the bivalents and by crossing over. So we're going to talk about independent assortment and crossing over and how both of those affect meiosis's ability to produce genetic variation in sexually reproducing organisms. So when we look holistically at meiosis, you will notice that there is a diploid parent cell. You'll also know that through 2 rounds of meiotic division, 2 rounds of cell division, there is 4 daughter cells produced, and all 4 of those daughter cells are haploid. So haploid meaning they contain a single set of chromosomes, diploid meaning they contain 2 pairs of chromosomes. Now our parent cells that will eventually go through meiosis contain 2 complete sets of chromosomes. You'll see in this picture there is a complete set of 3 blue chromosomes and a complete set of 3 pink chromosomes. The blue chromosomes represent our paternal chromosomes, and the pink chromosomes represent our maternal chromosomes. You get 1 complete set from your dad and 1 complete set from your mom. The complete set from dad comes through the sperm cell. The complete set from mom comes through the egg cell. Meiosis is the process by which we produce those gametes. And so what has to happen in meiosis is you start with that diploid cell that contains 2 sets of chromosomes. Again, diploid, we use the term 2N. N is our haploid number, and so if it is a diploid cell, it would be 2N, 2 complete sets. N would represent the complete set of chromosomes. A haploid cell has a nucleus which has 1 set of chromosomes. Again, we're going to use N, so essentially 1N. Haploid uses the term or symbol N. Meiosis is a reduction division as the parent cell is diploid and the daughter cells are haploid. Notice we are starting with a diploid parent cell and we are ending with haploid daughter cells. So we are reducing the number of chromosomes, which differs from mitosis. Remember when we talked through mitosis, mitosis is not a reduction division. If we start with a diploid cell, we're going to end with 2 diploid cells at the conclusion of mitosis. The whole purpose of mitosis was, remember, to ensure genetic continuity, meaning we're going to ensure that we're making a continuous cell that is genetically equivalent to the original parent cell. We use mitosis, remember, to grow as an organism. We use it to repair dead or damaged cells. And we also do it to ensure that the cells that we're producing are genetically identical to the cells we're replacing. Meiosis reduces the number of chromosomes by half in the nucleus of the daughter cells. And the whole reason we have to reduce the number of chromosomes is meiosis is all about producing gametes. And these final cells are called gametes. Now, in a sperm cell, these are the gametes that are made in males, and ovum or egg cells are the gametes that are made in females. Both gametes are different. They're different sizes, they're made for different reasons, but they both are made via meiosis, which is an important point to make when we talk holistically meiosis. Gametes possess half the number of chromosomes of the original parent cell. Okay, I keep harping on this, but gametes have to have half the number of chromosomes so that when they combine, it produces a diploid cell, or that zygote, that has the correct number of chromosomes to make a viable somatic cell going forward. So when we look at how haploid gametes come together, the father, remember, is male. He produces sperm cells through meiosis. The mom is female. She produces an egg through meiosis. In humans' case, again, this is showing human chromosomes. Not every sexually reproducing organism on the planet has the same number of chromosomes. We happen to have 46 total chromosomes in every somatic or body cell, diploid cell in our body. Since our diploid number equals 46, and we solve for n being 23, that tells us that our haploid number, n, has to be 23. And so there are 23 total chromosomes packaged into our egg cells, and there are 23 total chromosomes packaged into our sperm cells. When the sperm cell with its 23 comes together with an egg with its 23, it produces a fertilized egg known as a zygote that possesses 23 times 2, or 46 total chromosomes. Gametes must be haploid, otherwise the number of chromosomes possessed by each new generation would double. Again, we can't do that. Humans equal 46. We as a species, human, Homo sapien, has 46 total chromosomes, and we need 46 chromosomes in order to be human. If this chromosome number changes, the species changes. The human testes and ovaries, respectively, produce haploid sperm and egg cells through meiosis, so that when fertilization occurs, the zygotes will receive, again, 46 total chromosomes. So the father is going to produce these haploid gametes known as sperm cells in the testes, and the mom is going to produce these haploid gametes known as eggs in the ovaries. Both males and females will use meiosis in order to produce those haploid gametes. This is how the problem of changing the chromosome numbers is avoided. The chromosome number is preserved by the sexual life cycle. If we as an organism and as a species need to conserve our chromosome number, we have to first half our chromosome number to produce those gametes, so that when they come together they produce the viable diploid cell that will eventually produce a new organism. That is called the sexual life cycle. Now, if we look at meiosis holistically, you will notice that there are 2 rounds of cell division. So you'll notice that prophase 1, metaphase 1, anaphase 1, telophase 1, and then cytokinesis 1. All of these are going to be meiosis 1, and then all of these will be meiosis 2. Now, meiosis 1 is a reduction division. We're taking 46 total chromosomes and we're cutting it into 2 cells that have 23 chromosomes each. But you'll notice that the chromosomes are still replicated, meaning they are still in these X shapes. And so when we go through meiosis 2, we will take 23 X's and we'll cut them all in half, and we will start with 23 and end with 23, but the 23 we end with are not replicated chromosomes. They're not in the X shape. They're essentially half X's, or unduplicated chromosomes. So during the first round of segregation, the chromosome number is halved. We go from 46 to 23 X's, or 46 to 23 replicated chromosomes, duplicated chromosomes. And then meiosis 2 is we're going to take the sister chromatids, which are still in their X form, and we're going to separate them into 23 half chromosomes to finish out this meiotic division. So focusing on meiosis 1, zooming into meiosis 1, remember PMAT, prophase, metaphase, anaphase, telophase, P-M-A-T. It's the same order we saw in mitosis. Essentially the same things are happening, however there are some slight differences. Prophase 1, as the DNA becomes visible, the homologous chromosomes are attracted to each other and pair up or form the bivalent or the tetrad. So you'll notice that if this is chromosome number 1 and this is chromosome number 1, they come together and form a homologous pair, which we call a bivalent or a tetrad. Every single one of the homologous chromosome pairs is going to pair up. Chromosome pair number 1, chromosome pair number 2, chromosome pair number 3, chromosome pair number 4, 5, 6, all the way to chromosome pair 23. As those homologous chromosomes come together, they will begin to potentially cross over, meaning they're going to overlap in specific areas. And when paired and overlapping, they form a chiasma or a chiasmata, and that's when crossing over occurs. Now, crossing over is when they actually exchange or swap parts of their chromosomes. when they exchange sections of their chromosomes, it's called crossing over, and that helps to shuffle the alleles, or mixing of the alleles, which helps to increase genetic diversity and is one of the reasons why every single one of the gametes that is produced in both males and females, spermatogenesis and oogenesis, is different. Now, after crossing over takes place, we have the nuclear envelope dissolving, we have the spindle fibers producing, we have the centrioles migrating. Everything is equivalent to what happened in mitosis. However, when we get to metaphase 1, you'll notice that the pairs of chromosomes, or the tetrads, are lining up along that metaphase plate. So chromosomes line up randomly at the metaphase plate, ensuring random assortment of chromosomes. They do line up as a pair. Notice, though, that some of the pairs, the red chromosome is on top, and on this pair, the blue chromosome is on top. Now, on this middle pair, the blue could have been on top, and on this first pair, the blue could have been on top. That is the randomness of the way these chromosomes line up, and depending on how they line up, which is called independent assortment, dictates the way they're separated. So, because they lined up this way, the two reds will go to the top and the two blues will go to the bottom, and in that third pair, the blue will go to the top and the red will go to the bottom. Now, these could have lined up so that all three reds went to the top. It could have lined up so that all three reds went to the bottom. That is the randomness. That is the independent assortment. None of the pairs have any influence over the other pairs. The new daughter cells are haploid and contain half the number of chromosomes as the original parent cell. We have separated all of the homologous pairs. We have not separated the sister chromatids at this point. Once telophase happens, the cleavage furrow will form, the spindle fibers will break down, the centrioles are in their poles, they will eventually split the cytoplasm, and cytokinesis takes place. But because this is meiosis and we still possess our chromosomes in duplicated form, meaning we have two sister chromatids per chromosome, we are not going to run through an S phase, we're not going to run through another full cell cycle. We are just going to immediately go into meiosis II, which takes the two daughter cells that were produced by meiosis I and immediately runs them through meiosis II. And so now we have PMAT2, prophase, metaphase, anaphase, and telophase. Again, 2, 2, 2, and 2. And so the two daughter cells, which are haploid, by the way, this is a haploid cell because it has half the number of chromosomes we started with. The spindle fibers are going to start forming from the centrioles again. The centrioles had to duplicate. The nuclear envelope never really formed, and so it's just kind of hanging out, ready to form the new nuclear membranes when the cell divides again. And so as we start to look at meiosis II details, you'll notice that since the chromosomes never fully decondensed, they are not in their homologous pairs. We have separated those homologous pairs, and so the chromosomes still contained their swapped alleles. Notice that crossing over, which happened in meiosis I, still has the swapped parts to the arms. That, again, helps to ensure genetic diversity. As we continue through meiosis, the chromosomes line up at the metaphase plate in random orientation. Again, it doesn't matter if this full red one lines up on the left side or it reverses and the full red one's over here. The way they line up is completely random. Again, independent assortment and segregation states that the way they line up randomly is going to dictate how the alleles are separated. Anaphase II, the sister chromatids separate and are now individual chromosomes. So at this point, we are not referring to them as sister chromatids. They are unduplicated chromosomes. They will move towards the poles. The spindle fibers between them will elongate. The spindle fibers connected to the centromeres will shorten. And at the conclusion of anaphase II, 4 haploid gametes are produced and are all genetically unique. Notice that every single one of the cells has a different combination of alleles, and cytokinesis will take place, resulting in 4 haploid genetically unique cells. So, when meiosis happens correctly, this is what the result is. So normal meiotic division results through 2 rounds of meiosis, results in 4 genetically unique but genetically viable gametes. Meiosis usually produces viable haploid gametes. Again, we started with a diploid cell and we resulted in 4 genetically viable unique gametes. If, however, meiosis doesn't happen correctly, we can result in genetic disorders, and Down syndrome is one of those disorders. So, when chromosome pairs fail to separate, it's called non-disjunction. And non-disjunction can happen in meiosis I, and non-disjunction can happen in meiosis II. If non-disjunction happens in meiosis I, and that pair fails to separate, that entire pair will go to one of the two daughter cells, and then they will divide through meiosis II to produce gametes like they should. However, because the pair failed to separate, you end up with two gametes that have an additional chromosome, and you have two gametes that are completely missing a chromosome. These gametes will result in a trisomy because they contain two sets of that particular chromosome. And let's say if these were egg cells and we had a sperm cell that had its normal chromosomes, they would be added to this egg, which would result in a pair of the short ones and three of those long ones, which again, that's what trisomy means, three copies of a particular chromosome. If non-disjunction happens in meiosis II, you'll see that the sister chromatids at this point fail to separate. That sends these, after meiosis II, both to this particular gamete, which would result in a trisomy. This gamete would be missing one, and you would have two normal gametes as the result of normal meiotic division through this right side. Non-disjunction and a potential future trisomy results in a disorder known as Down syndrome. Okay, Down syndrome is a genetic disorder, is a physical and cognitive disability. So one of the things that contributes to the greatest amount of genetic variation within meiotic division is the process of crossing over. And so if we look at crossing over, homologous chromosomes pair during prophase I and form those tetrads or bivalents, meaning the paternal chromosome, which is the chromosome you received from your dad, and the maternal chromosome, the chromosome you received from your mom, join up. So this would be the paternal chromosome number 1, this would be the maternal chromosome number 1. They both have the same genes, but obviously can have different alleles. In this case, the paternal chromosome has the dominant A and B, and the maternal chromosome has the recessive A and B. While paired, homologous chromosomes can form a chiasma and exchange portions of their arms. That is crossing over. So when they overlap, the location of the overlap is the chiasma. Okay, so they physically cross their arms, and then when they cross their arms, they cut the arms off of each of their chromosome and exchange them so that they now possess alleles that they originally didn't have. So when we look at the original paternal chromosome, this original paternal chromosome had a dominant AB linked, and the original maternal chromosome has a recessive AB linked. And when I mean linked, the little a and little b are linked to the same chromosome. They are located on the same sister chromatid. And so this individual would have the genotype big A, big B, little b. And if you remember from our inheritance talks, that would be an individual that is heterozygous for the gene A and gene B. When this individual, because they are linked, produces their gametes, the only gamete options that this individual will produce without crossing over is an AB linked and an AB linked. It is impossible without crossing over for this individual to produce a big A and little b or a little a and a big B. So these are the only two options. In fact, 50% of this person's gametes will be this and 50% will be this. Now, how does crossing over affect that? If we crossed over between these two sister chromatids within this tetrad, you get this situation where you have one sister chromatid that is big A, big B, so big A, big B linked. This sister chromatid now contains big A, little b linked. This chromatid now has little a, big B linked, and this chromatid is little a, little b linked. And what happens from this one crossover event is that now there are 4 gamete options, and each of them have an equal 25% chance. So we go from having only 2 options in our gametes to having 4 options, and so one crossover between one homologous pair doubles the number of gamete options we have through meiosis. So crossing over produces recombinant chromatids that contain portions of maternal and paternal DNA. This one and this one are what we call the recombinants, and those are basically a mixing of the maternal and paternal alleles, meaning we mixed up big A and little b and little a and big B. Those did not happen or would not have been possible before this crossover event. And so now if we look at meiosis from an independent assortment standpoint, independent assortment is the random orientation of the chromosomes during metaphase 1 of meiosis. So when we look at this particular image, this is showing two different outcomes for the same starting diploid cell. So this individual cell would be a diploid cell. Notice there are two complete sets of chromosomes. You have a blue set and a red set, and both of them are identical. If this option on the left happens, or if this is our meiotic division, and in meiosis 1, both red chromosomes line up on the right, and both blue chromosomes line up on the left, you go through 2 rounds of meiosis, meiosis 1, meiosis 2, and you get 50% little r, little y, and 50% big R, big Y. Those are the only 2 options. But if you take the same genotype, the same starting diploid cell, and you simply line up the chromosomes differently, you get an entirely different outcome. So here we have the big R, or the red, on the right with chromosome pair number 1, and you have the red one now on the left with chromosome pair number 2. Notice that we just swapped the orientation of the second pair. When you go through meiosis 1 and then through meiosis 2, now based on the way they lined up and the way they separate, you get 50% little r big Y and 50% big R little y. These 2 options are different than these 2 options, and the only reason they're different, meiosis is the same. The way they go through their reduction division is the same. All of the steps of meiosis is the same. Metaphase 1, metaphase 2, all of that is the same. The only thing that differs is the way the chromosomes lined up, which resulted in completely different outcomes, which, again, is one of the reasons why meiosis produces genetically unique cells and why meiosis and the production of gametes leads to genetic variation within sexually reproducing organisms. That's it for this particular video. If you learned something, give it a thumbs up, leave a comment. If you have any questions, subscribe to the channel. We'll see ya.